Good morning, everyone. Good morning, Janak. Good morning, Vikesh. Just a minute. Just 1 minute. Okay, let's begin with the class now. So today is the third day of our crash course for your ARS net examination, right? So make sure that if you are targeting out the ARS net examination, the upcoming ARS net examination, you are following up with these questions, with these topics, because it will help you or it will guide you to go through the topics in a specific pattern, I would say, because ARS net questions are majorly very static and they are very to the pointers. So I've tried to add on as many topics as possible for your examination from the various sources, like from whichever source it was possible, right? So just a reminder, we are continuing with our focus batch on our application, right? If you are preparing for the CSIR December 2025 examination, so this particular course is going to help you out. It's a all-in-one course. It's a complete detailed batch which includes live plus recorded classes, hard copy notes, PYQ hard copy, and right now we have our Freedom Day sale also ongoing, right? We have our sale ongoing. The Independence Day sale is also ongoing right now. So if you wish to enroll in any of the course, you can enroll with the discounted prices, right? So make sure that if you wish to enroll, you can use this particular window of 12th to 17th. It's 14th today, so you still have 4 days in hand. If you wish to enroll, you can enroll accordingly, right? Now if I talk about the topic today that we are going to discuss in, so the very first topic that we are going to discuss today is your plant disease mechanism. So it is a common topic in a number of different units, right? In different subjects, like for example in plant biochemistry it is also there, in plant physiology also. If you are like giving the examination in plant physiology, plant pathology, plant biochemistry, so this entire topic is going to help you, right? The plant disease management, in this the pathogenesis, resistance and the PR protein, all these topics are something that are going to that we are going to cover up today, right? Good morning everyone, good morning. Okay, so if I talk about that, what is a disease or what is a plant disease all about? So what is it? Any disturbance of normal, like how do we define a disease in general? Disease means what? Disease means that if any normal functionality is getting hindered, or if any normal functionality is getting altered altogether in the normal functionality of the individual, that is what a disease is. Like if I say that I am having a disease, that means one of the functionality or one of the mechanism in my body is not functioning properly, right? So that is what a disease is. So if a plant disease I am talking about, so plant disease means any disturbance of normal physiological and biochemical function in a plant that is caused by the biotic or abiotic factor. Obviously, the factor which can help out, the factor which can cause the disease, anything that can cause the disease, it can be a biotic factor, it can be a abiotic factor, right? It can be a living pathogen, it can be the non-living factors like any specific very low temperature, very high temperature, very high heat, or anything could be there which is going to cause the disturbance or which is going to cause the functionality to not work in a proper orientation, right? Now, what are the various major plant pathogens that we have? So, we have fungi, we have bacteria, we have viruses and viroids, phytoplasms and nematodes. So, fungi is, example is Puccinia, Fusarium and Alternaria, right? Then in the bacteria, Xanthomonas, Pseudomonas. Virus and viroids, TMV, CMV, PST, VD. Then phytoplasma, so little leaf of the brinjal. Then nematodes, root knot nematode. So all these are the various major plant pathogens that we talk about, right? So what is a plant disease we have discussed, and it will lead to showcasing of the symptoms and the various economic yield losses that we have in, right? Economic yield loss means that if any plant which is commercially being grown in, let's say wheat, let's say rice, so if there occurs any kind of disease in the entire crop or in the entire field of the area, so what will happen? The economic yield loss will also be therein, right? So plant disease is what? Any disturbance in the normal functionality which leads to visible symptoms and economic yield loss in the individuals, right? Then economic importance of the your plant disease or economic importance of the plant altogether is that it leads to, or it causes what? It causes annual global crop loss of more than 20 to 30%, which is a very high amount of loss in the individuals first, and it is going to cause a number of diseases, like it is going to cause a loss of a lot of like money, labor and all those things, right? It reduces the yield, market value get reduced, and the seed quality also get reduced because the entire crop is getting impacted, right? So the global loss will also be therein. Example is your wheat rust epidemics, late blight famine, etc. Then what is the disease triangle? This has been asked in the examination before. Disease triangle means that a disease will occur in 3 scenarios, and when these 3 scenarios are therein, all these 3 scenarios will be like getting summed up together and the disease will be there. For example, first one is susceptible host, that is whatever host is there, it is susceptible in nature, means the pathogen can easily attack on it. It could be weak, it can be young, it can be having some other disease already, right? Then virulent pathogens, that is whatever pathogen is attacking on, it is having a very high virulence activity. Then favorable environment, and then the favorable environment, like that is the environment for this particular pathogen, whatever is causing the disease, is favorable in that, like the pathogen can easily grow in there, right? If these 3 things merge in together, you will have your final disease to be showcased in the individual, right? So susceptible host, virulent pathogen and favorable environmental coincides. And why do we have to study this pathogenesis, and why do we have to study the resistance altogether? So for sustainable development, for breeding programs, and for predicting the disease outbreaks altogether. 3 major things. Why are we even studying the pathogenesis and resistance? So sustainable management, that is reducing the pesticide dependency, then guiding the breeding program, guides the breeding program for the durable resistance, and then essential for predicting the disease outbreak and developing the IPM parasites, right? So all these are the reasons why pathogenesis and resistance is therein, right? Or why do we study about it, right? Did you understand everything till here, everyone? Yes. Everything clear to everyone till here? If this parts get clear, we'll discuss about the further parts. We'll discuss questions as well, so make sure that you're there in the class till the end. Yes. Okay. No, no, it is like completely general topics that I'm taking about, so that majority of the students can get the help out of it. Agriculture or agricultural microbiology, few topics you can take it from here. So you have to check your own syllabus, because there are so many subjects in entire ARS net, and it is very difficult for any of the platform to cater all the topics all together. So I have tried to jot down few of them, few of the topics, and then I am taking up over here. Okay, so if this topic is there in your segment, you can take it up. Okay. Then what is pathogenesis, the definition and the concept. So pathogenesis is what? It is a complete sequence of events from the arrival of pathogen inoculum to the development of the viable disease symptom. That is, from the time your viable pathogen inoculum means the initiator segment with which the pathogen can easily develop in, the initial colony of the pathogen. That is what inoculum is. So pathogen inoculum, from the like sequence of pathogen inoculum till the development of visible disease symptoms, we have the entire complete sequence of event known as the pathogenesis part. Okay, what does it include? It includes infection, it includes colonization, it includes symptom development, it includes reproduction, right? So starting from infection, then increase in the number, that is colonization, then symptoms, and then the reproduction, all these things. Then what are the key aspects? First one, it involves the pathogen strategies plus the host responses, right? Pathogen strategies means the enzymes, toxin effectors, and the host response, that includes the defense activation, resistance gene, all these things. Resistance gene are specifically known as your PR genes, right? Pathogenesis related genes. Then dynamic process, so it is influenced by the host genome, genotype, pathogen virulence, and the environmental conditions. What are the importance that we talk about? So it helps to identify critical control points for the management. It explains the host specificity, and it is used for the basic understanding of disease resistance breeding, right? So what all are the functionalities or the importance of the plant pathology? So it is development or helps in the critical control pointers, specifying the host specificity, and for the basic understanding of the disease resistance breeding. Now what are the steps? Now this can again be a good question in your prep, in your ARS net, that what are the various steps in the pathogenesis. Starts with inoculation, then we have attachment, then we have penetration, then we have infection, then we have invasion, colonization, symptom development, reproduction, dissemination, and survival. 10 steps are there. Starts with the inoculation, that is when there occurs arrival of the pathogen propagules, right? Then attachment, so adhesion of the host surface, adhesion to the host surface via mucilage, adhesives, aperosias, anything. Then penetration, that is after the attachment, penetration into the plant body. It can be via direct natural openings like stomatas, lenticels. It can also be wounds, direct enzymatic degradation, etc. Then infection, so establishment of a parasitic relationship and initiation of the nutrient intake, because only when there is relationship between the host and the parasite, then only the nutrient uptake can begin, and there need to be a physical relationship between the two for a proper exchange of the content, right? Then invasion, so local and systemic spread through the intercellular spaces and or the vascular tissue. Then colonization, so multiplication and establishment within the host tissues, because after the everything has been completed, now the number of pathogens need to be increased to increase the intensity of the reaction and increase the intensity of the infection and everything. Then reproduction, so formation of pores, etc. Then dissemination, that is spread to the new host by wind, water, insects, tools, seeds, etc. Then survival is persistence in the soil, crop residue, seed, alternative hosts during the adverse condition. All these things are going to help out in the, or all of these things are going to cause the pathogenesis in a sequential manner. I've just told that I am taking up general topics which are common in a number of different subjects altogether. Okay, I'm not going to take any specific subject as such, because there are ample, like I guess more than 50 subjects from which the ARS net examination will be conducted. So it is impossible for any platform, not just me but any platform, to conduct or to take up class for every single one. You can check, I think agricultural biotechnology will be having this pathogenesis topic, so you can utilize this particular segment over there. So I am just taking some general topics which can be there or which can be utilized up. Okay, what are the factors for pathogenesis? We have cell wall degrading enzymes, we have the toxins, we have the growth regulators, we have effector protein, polysaccharides, cytophores, then cell wall fragments, and then suppressors. So all these are the various factors which are going to cause the pathogenesis. Okay, for example, the first one, that is cell wall degrading enzymes such as cutinase, pectinase, cellulase, hemicellulase, they all break up the physical barrier in the plant. Then host specific toxins like Victorin, T toxin, non-specific toxins like TAP toxin, fusaric acid, all these things. So you make sure that you remember all these things, right? What are the various defense mechanisms that the plant showcases? So first is the structural barriers, that is thick cuticles, waxes layer, etc. Anatomical defenses, that is trichomes, stomatal closure, abscission of infected organs, etc. Preformed chemical defenses, antimicrobial, phytoanticipants like saponin, tannins, etc. Induced structural defensin, induced chemical defenses, then oxidative burst, hypersensitive response, that is HR, signaling pathways, salicylic acid, jasmonic acid, all these things. Signaling pathway, RNA silencing, and the systemic responses. So what are the various preferred defense mechanisms? All these are the various defense mechanisms that would be there, right? Starts with the structural barriers, then anatomical defenses, preformed chemical defenses, induced structural defenses, oxidative burst like rapid ROS production, reactive oxygen species production at the infection site, then hypersensitive response, localized cell death to limit the pathogen spread, signaling pathways, that is salicylic acid, just like when we learn about plant hormones and specifically your jasmonic acid and salicylic acid, over there we have these signaling pathways where we learn about that these are the ones that help out in the plant defense mechanism if a plant is getting infected by a pathogen. Then systemic response, so SAR and ISR provide long lasting immunity. Systemic acquired responses, SAR, right? Then okay, now you have this part. Now you can take a screenshot of this entire segment, this table. It is going to help you like anything. That host, what are the key features for the host, what are the various examples for the host, what ICAR exam tips you can take up, right? Then pathogen, what are the key features, example, questions, what type of questions could be therein. So first thing is your host. So it must be susceptible, resistance genes absent or ineffective, influenced by age and physiology, right? Example is young seedlings more susceptible to damping of, drought stressed plants more prone to wilt, and it often asks stage specific susceptibility examples. Then pathogen, so it must be virulent, high inoculum potential, effective reproduction and dispersal, like Phytoplethora infestis, Puccinia graminis. Question may involve pathogen race and the virulence. Then environment, condition must be favorable for the infection, that is for the pathogen and the pathogen growth, temperature, humidity, pH, wind, etc. Late blight needs cool, void, moist weather. So that whatever the what do I call, infection need to be there, whatever the pathogen need to be there in over there, that pathogen's condition is something that we are going to, that need to be prevailing in the environment for the same. Common diagram questions, label the triangle correctly, these type of questions are asked in. Then interaction outcome, if all the three factors are present, disease develop, anyone absent, no disease. I have just told you disease resistance, pathogenicity and survival over here in the very beginning, right? So that particular segment is over here. Then compatible interaction leads to infection, incompatible leads to resistance. Then missing factor scenarios often asked in, out of the three triangle, from there the question get asked from here. Then management link, so altering any one of the factor can prevent the disease, adjusting the planting date, resistance cultivator and aggregation methods, and also the questions are asked majorly from your disease forecasting and IPM questions. Okay, so remember about this, these slides have been made with utmost care in the best content possible, so make sure that you remember all of these things. Yes, now we'll discuss few questions. Yes, understood everyone till here? If you want to take a screenshot, you can take a screenshot as well. Yes, understood? Done? Okay. Then we have the next part. See over here. Okay. We'll discuss few questions now. Let me see if you can mark some answers or not. Okay. First is the gene for gene hypothesis was first proposed based on the studies in. Gene for gene hypothesis was first proposed based on the studies in rice, flax, wheat, tomato. Try to mark the answer for this. Let me see if you can mark it. This one, all these questions will be very much factual. Gene for gene hypothesis was first proposed based on the studies in rice, flax, wheat, tomato. Yes, mark the answer for this. Okay, none of you have marked it. Answer for this is option number 2. The gene for gene hypothesis was first proposed based upon the studies in flax, that is this one. Okay, say these type of questions are only asked, and if you think the questions would be from simple areas, no, the questions will be from these tricky areas only. Which PR protein group contains beta 1,3 glucanase? Can you try this one? Which PR protein, pathogenesis related protein group, contain beta 1,3 glucanases? PR1, PR2, 5 and 10. Let's see. Answer is PR2. So PR2 contains beta 1,3 glucanases, the enzymes that degrade the fungal cell wall glucans, weakening the pathogens, right? So answer for this is option number 2, PR2, that is beta 1,3 glucanases is in the PR2, pathogenesis related protein 2. Thaumatin like proteins belong to which group? This has been asked in the examination before, as it is same question. Thaumatin like proteins belong to which group of PR proteins? PR3, PR5, 7 and 9. Think about this. Let's see. Thaumatin like protein belongs to PR5. You have to remember all these parts. Thaumatin like proteins belong to which part, which group? So it is the PR5. They cause membrane permeabilization in the fungi, right? So B is the answer for this particular question. It takes a lot of energy to curate these classes. Okay, so if you don't want me to take up these sessions, please let me know. I have a new series already waiting. I was just making sure that few students get the help for the ARS net a bit, but I can't see much of the response by the students. So this would be the last class for the ARS net, and from Monday I am starting off with my CSIR series. Okay, timing would be similar, 11 AM, or it might change a bit. I'll let you know. Otherwise I'll be taking up the sessions for the CSIR and negate now, and not for the ARS net anymore. So this is the last question also. In the disease triangle, removal of which factor always prevent the disease development? Which factor can always prevent the disease development? So any of one of them, either the host or the pathogen or the environment, can cause the disease development to be prevented, because all the three things need to be there in pathogen, host and the environment, right? So answer for this is option number D, right? So this is about for this particular session. I hope all the things are clear to everyone. If you want to have this particular segment, you can take it from the Instagram, from the application. I'll upload this particular PDF over there, right? So that's about it. Let's meet in the next class. And just a reminder, we have a freedom sale ongoing from 12th till 17th. So if you wish to enroll in any of the course, there is flat 25% offer available. We have our handbooks, we have our previous year question paper compilation. So whatever is your requirement, you can enroll in accordingly. Okay, so that's about it. All the best, good luck, take care everyone.